Hello, everyone. First of all, I'd like to thank the organizing committee for inviting me to give a speech in this very honorable uh, event. And so I'm going to talk about the personalized medicine with the other saying pharmacogenetics or precision medicine. And almost at the end of the speech, I'm going to give some examples of AI be used in the therapeutic medication monitoring. So let's start with the pharmacogenetics. Actually, pharmacogenetics, as everybody knows, um, is the hybrid and translational science, and it was actually brought up for a couple of decades. And in the pharmacogenetics, actually, we are looking at the pupil's genetical status, and we are asking the physicians to give right prescription without any side effect of them. Actually, Üsküdar University is based in Istanbul, and this university was established by the Brain Hospital of Turkey. And this brain hospital is the third one in the world, and the first one was established in the United States, second one in Hamburg in Germany, and the third one in Istanbul in Turkey. And that's why we are dealing with the patients who are suffering from neuropsychiatric diseases. So we established our pharmacogenetics lab in 2013. Actually, the university started education in 2012. Then we established the lab. And later on, now we are still organizing to establish some panels to give the right prescription, right time, without side effect and without toxicity on them. Let's start actually pharmacogenetics. What is the pharmacogenetics, and when or what is it is a root back to history. In fact, as you know, everything almost has started with the elucidating the structure of the DNA, and it was published in 25 April 1953 by James Watson and Francis Crick and the other colleagues. And after this, actually, we started the Human Genome Project in 1990. In fact, people were assuming to complete the project in 2015 to 2020. But as you know, all scientists, and as we are studying some issues and new techniques we are developing, and therefore, the expectation of the Human Genome Project's results to be announced 2015. Actually, it was done 2003. So, it is very quick results we reach. So by using the genetical data from the Human Genome Project, and we try to apply genetical data used in the treatment of the people. As everybody knows that, everyone has different type of the genotype. So these genotypes are affecting the human-drug interaction all the time. And this is actually one of the translational science. Nowadays, artificial intelligence involved in these kind of science. Then now we are almost starting to use artificial intelligence to classify main disorders. And actually, when we look at the drug metabolites, intermediate metabolites in the body, even we can subclassify the major diseases. And I'm going to a little bit later talk about it. And the Human Genome Project paved the way, and in addition, it is called as precision medicine, biological engineering, computational and mathematical biology, systems biology, and translation research informatics. So one of them is pharmacogenetics. And the Time published one of the issue related with the personalized medicine or precision medicine. And after that, Clinical Pharmacology and Therapeutics Journal put a picture related with the personalized medicine. And why we should use personalized medicine? Because personalized medicine give us some benefits. And the first of all, effective drug formulation, improve safety of the drug, increase potential of drug, improve patient's care and compliance, and right selection of the drug, and improve drug elimination. Actually, it is really important. When we look at the economical benefit of the precision medicine or pharmacogenetics, actually, in the United States, they did research, and in this research, the study used a comprehensive medication management system enriched with the pharmacogenomics and reported a $32 million reduction in direct medical cost during the first 32 months in a cohort of approximately 5,000 personalized genomics X-guided patients. And look at that one. Actually, it's only 5,000 people, and then the benefit is $32 million. And what is the new in the pharmacogenetics? Actually, as the traditional medicine or the medicine at this time actually use try and error method because the number of the patients are very huge and the number of the doctors are very less. I mean, when they compare with the patient and the doctor ratio. Therefore, they are just looking at you and maybe doing something, and sometimes without asking any lab work, then they are trying to diagnose your disease, and they are giving a drug. Then you are going home. But maybe starting from today, and it will be in the tomorrow, they are going to have a look at our genes related with the the drug metabolizing enzymes, and then they are going to give us a really perfect medication. In fact, try and error and personalized treatments are really different. And in the try and error technique, doctors are prescribing the same protocol for all individuals. And in contrast, in the personalized medicine, the protocol is applied according to individuals' genetic characteristics. So if you look at this figure, then in the standard approach or one fit all concept, the result is showing that the treatment effectiveness is around 20%, and 80% of the medication and the time are wasting. It is really interesting. But if you give the treatment protocol for every single one regarding the genetical features, and then the effectiveness of the treatment is going to be 99.5%. Actually, there are two terms related with the pharmacogenetics and the pharmacogenomics. In the pharmacogenomics, actually, sometimes we need to have a look at the whole genome sequencing. But if we look at, and if we are dealing with the only couple of genes, then it is called as pharmacogenetics. The aim of the pharmacogenetics actually for each patient, the most appropriate drug and the most appropriate dose, and to develop personalized treatment approaches based on the patient's genetic profile. And please bear in your mind that worldwide misuse of medication is the fourth leading cause of death. It is really amazing, and it is really interesting result. Pharmacogenetics actually includes the concepts of the pharmacokinetics and pharmacodynamics. I'm not going to explain to your honorable audience the meaning of that, but maybe the concept of these, these words are what our body does to the drugs we took. And the second one is what the drugs are making to our body. It is really interesting and simple way to have a look at. And the goal of pharmacogenetics or pharmacogenomics is maximizing the drug efficacy and minimizing drug toxicity, predicting patients who will respond to treatment or the protocol, and supporting the development of new drugs. Actually, it is one of the another important aim and the goals of the pharmacokinetics and pharmacogenomics. Actually, as you know, there are some factors associated with the drug response and adverse effects, and these are exemplified in this, as shown in this figure. In the pharmacogenetics, actually, we are really interested in with the genetic differences. As you know, there are some variations in the population, and if these variations are occurring with the mutations in our body. So, as you know, there are different type of the mutations, but we are interested in the point mutations in this case. If the mutation is, the number of the mutation is, or the frequency of the mutation is equal or over the 1% of the population, we are calling as, polymorphism. Actually, maybe we are softening the mutation, but whatever it is because of the mutation. And we are really interested in the single nucleotide polymorphism in the pharmacogenetics. In this case, do they are really functional, or can we reverse these activity or can we reverse the inhibited genes to work again? As you know, in the genetical code, sometimes only one nucleotide is changing the functional activity of the proteins synthesized. So, as both in the sickle cell anemia and the thalassemia, there are only one nucleotide changing, and in one case, in the sickle cell anemia, amino acid sequence is changing, and then the globulin doesn't affect properly and precipitate in the red blood cells, and that's why they are changing the red blood cells shape. On the other hand, in the thalassemia, again, only one nucleotide is changing, and then the codon, genomic codon, is becoming stop codon. Then there is no further translation in the protein biosynthesis. So these are really big problem for us. In the genetical polymorphism, actually, it could be related with the pharmacokinetics and for the pharmacodynamics as well. And these polymorphic characters, for example, related with the pharmacokinetics, they could be transporters, they could be plasma proteins, and they could be metabolic, velocity or metabolic activities. And related with the pharmacodynamics, and these polymorphic features are affecting the structure of the receptors, ion channels, enzymes, and immune molecules as well. And because of that, the changes, as you know, this is the the differences of all of us actually, and even in the phenotypic polymorphism, and you can see the butterflies and these insects. So, which genes and which gene products are really important for the pharmacogenetics? And these are metabolizing enzymes, actually. And as you know, there are two types of the enzymes in the metabolism, and these are phase 1 enzymes, and this is the phase 2 enzymes. And if there is any mutation which affecting the activity of the protein, then it is really giving us really big problems. And these enzymes, mainly actually cytochrome P450 enzymes, and then until now, 57 enzymes are responsible for that. And most important, the CYP450 family includes many polymorphic genes, actually. And these genes are affecting our metabolism or our metabolism related with the drugs we use. And CYP-mediated metabolism of the different drugs, and you can see CYP2D6, CYP2C9, CYP2C19, etc. And you can see the which kind of molecules are metabolized by these enzymes. Among these, actually, mainly we focused on the, of course, you will see, I'm going to explain how many genes and how many polymorphic characteristics we are looking for. And then I'm going to give example with the CYP2D6. And actually, it is primarily active in the liver and important enzyme across the broad range of the drug metabolism for antidepressants and antihypertensives and chemotherapeutics. Actually, in the neuropsychiatric disorders in humans, we are looking at these enzymes. Let's have a look, the one example with the CYP2D6 actually. And in the psychotropic drugs and pharmacokinetics related with these enzymes and pharmacodynamics are related with the, for example, serotonin transporter and serotonin 2A receptor. And in the ADHD disease and pharmacokinetics is related with this 2D6 and pharmacodynamics actually it is adrenergic receptor and catecholamidyl and when we look at the analgesics again CYP2D6 related with the pharmacokinetics and when we look at the pharmacodynamics opioid receptor is related with this polymorphism. Actually, because of these kind of the polymorphic characteristics, and people are classified into 4 groups actually, and if the enzyme is metabolizing very quickly, and these are called as ultra rapid metabolizers, and the extensive and intermediate are very good actually, they are metabolizing very well. But when we come to the poor metabolizer, then it is another problem. Then they cannot metabolize the drug. So, if you the same drug is given, all the time by increasing the dose, then we are actually poisoning the people. And this is a very good example, actually. The same drug and 3 people with the 3 different type of the metabolizing capacity. And then, for example, in the patient 1, and when venlafaxine, actually, it is given 75 milligram to them, and full remission you will get. And their CYP2D6 actually extensive metabolizer. So they are really metabolizing this drug very well. Then their result is full remission. In the second patients actually, and he is taking double of the patient 1, and then they have some really side effect related with the drug, and there is no response. And so his CYP2D6 is poor metabolizer. When we come to the patient 3 and the same drug and 4 times bigger than the patient 1 taking every day and no side effect, but no response again. And because her CYP2D6 is ultra metabolizer. Therefore, we have to have a look at these kind of things. And this figure actually showing the, the metabolizer types and what's happening in the body. So, when we come to the pharmacodynamics, genes and the medication, actually, in the serotonin transporter, examples of some affected medication, as you see here, escitalopram, sertraline, and fluoxetine. So you can have a look all these things then. And these are really related with these kind of the proteins. So let's have a look the example of that. And in this case, serotonin transporter is the one of the big example in front of us. So serotonin transporter is encoded by SLC6A4 gene, and its function is serotonin uptake by presynaptic neurons. Actually, and these are the receptors, and then the proteins on the surface of the membrane, and they are interacting with the serotonin. And people are classified into two groups, actually. Maybe three groups, but this gene has two variants, and the short and long one. And the two variants differ by a 44 base pair insertion or deletion. So the short allele is associated with lower transcription, which results in fewer active SSRI binding sites. And the short allele is associated with a lower treatment response after SSRI administration. And look at this one, and if the gene is long or long S or SS, then this showing the long and long S, a little bit long S, and there are lots of actually transported protein on the surface of the neuron. So when it is SS, as you see, there are very small amount of the protein, will affect the serotonin, or will interact the serotonin. So, in the especially the resistant depression problems, and people are becoming really SS genotype. It is really interesting. And what we are doing in our laboratory, and these genes, and looking at the genotyping of these one, and actually you can see the how many areas we are looking for these kind of things, and then we are reporting to the psychiatrist or neurologist to give the prescription for the patients, which should affect almost 100% with no side effect. And ADHD panel and AD panel, Alzheimer's disease, and this is the susceptibility, and this is the pharmacogenetics and pharmacodynamics of the Alzheimer's disease, and the tests, as you see here, and we are doing, and we are analyzing by using these equipment. So one of the most important thing is the therapeutic drug monitoring for these patients, because they either forget to take medicine, or they have some idea in their mind that the people are trying to poison them. And so, they are not taking the drug regularly. And therefore, to have a look at if the drug is working with the his body, then we have to monitor the drug level in the blood. So actually, we are looking at their drug level and the intermediate metabolites in the blood. Then we are keeping now lots of data we have, and we are going to use them in the deep learning machine system. Therefore, one of my colleague, she developed an algorithm that by looking at the intermediary drugs or metabolites in the blood. She realized that when we took all the depression people and look at their drug monitoring results, and then we are now subclassified depression population now. And we are using this algorithm with other diseases as well. And so artificial intelligence is really going to be in everywhere in our daily life. Therefore, as a people like us who are studying on neuroscience, so we have to find out, some gates to introduce our results to the artificial intelligence. And I'd like to thank you for listening to me. And this is our lab when we established in the first time. And now these are actually undergraduates and postgraduate students doing their graduation thesis and the master degree thesis. And there are lots of people, and they are studying and working on this, and every day we are increasing our panels. Thank you very much for your patience, and I wish you very successful symposium. Thank you.